Welcome. Today we're diving into a breakthrough from Memorial Sloan Kettering Cancer Center, where researchers have found a way to hack the internal clocks of neurons. This discovery could dramatically speed up the study of neurological diseases like Parkinson's, Alzheimer's, and autism. Traditionally, growing human neurons in the lab takes months, mirroring the slow pace of human brain development. But what if we could accelerate this process? Let's explore how scientists are making this possible and what it means for the future of brain research. Human neurons mature slowly, which is great for our complex cognitive abilities but poses a big challenge for scientists. When researchers try to study neurodegenerative or neurodevelopmental diseases in the lab, they have to wait months for stem cell-derived neurons to reach an adult-like state. This slow timeline makes it difficult to model diseases and test new treatments efficiently. The need for a faster way to grow mature neurons has been a major obstacle in neuroscience research. Journey begins with human pluripotent stem cells. These remarkable cells can develop into many different types of cells, including neurons. In the lab, scientists use them to create nerve cells for research. However, the process is painstakingly slow, as these stem cell-derived neurons follow the same developmental timeline as those in the human body. This means months of waiting before experiments can even begin. Team led by Dr. Lawrence Studer at MSK has uncovered a way to hack the internal clocks of neurons. Their research revealed that an epigenetic barrier inside stem cells acts as a brake, slowing down neuron maturation. By inhibiting this barrier, scientists can speed up the development of neurons, bringing them to an adult-like state much faster. This breakthrough opens new doors for studying brain diseases in the lab. Let's break down the concept of the epigenetic barrier. Epigenetics refers to changes in gene expression that don't alter the DNA sequence itself. In this case, the barrier is a set of chemical modifications that slow down how quickly stem cells mature into neurons. By targeting these modifications, researchers can effectively remove the brakes and let neurons develop more rapidly. Dr. Gabriel Seisri, a key researcher in the study, noticed that neurons in mice develop much faster than in humans. In mice, neuron maturation takes hours or days, while in humans it takes weeks or months. The team found that the epigenetic barrier is present at higher levels in human neurons compared to mouse neurons, helping explain why our brain cells take so much longer to mature. Might seem surprising that these discoveries come from a cancer center, but MSK has a long tradition of basic science research. Understanding how cells develop and age is crucial for both cancer and degenerative disease research. Many advances in cancer treatment, like immunotherapy and CAR T-cell therapy, have roots in basic research just like this. Basic research is all about building a fundamental understanding of biology. About half of the NIH budget supports this kind of work, and it's the foundation for most new medical breakthroughs. The discoveries made in labs like MSK's often take years to translate into treatments, but they're essential for progress in medicine. A second study, MSK researchers developed a chemical cocktail called GE-N-Tony-K. This mix of four chemicals both suppresses the epigenetic barrier and stimulates factors that promote neuron maturation. With GE-N-Tony-K, scientists can bring neurons to an adult-like state much faster, making lab studies more efficient and opening new possibilities for disease modeling. GE-N-Tony-K cocktail doesn't just work on neurons. Researchers found it also accelerates the development of other cell types derived from stem cells, such as melanocytes, pigment cells, and pancreatic beta cells, endocrine cells. This suggests the approach could be useful for a wide range of biomedical research, not just neuroscience. Speeding up neuron maturation in the lab is a game changer for studying diseases like autism, which involve problems with how neurons connect and communicate. With faster models, scientists can better understand these disorders and test new therapies more quickly. The GE-N-Tony-K approach provides a valuable tool for researchers worldwide. While these advances are exciting, challenges remain. Diseases like Parkinson's typically appear late in life, often when a person is 60 or older. To model these conditions accurately, researchers need to find ways to not only mature neurons but also age them in the lab. This is an ongoing area of research, and scientists are working to develop models that reflect the aging process. This groundbreaking work was led by Dr. Lawrence Studer, Dr. Gabriel Seisri, and graduate students Emiliano Hergenritter and Andrew Minotti, among others. Their collaborative efforts at MSK and Weill Cornell Medicine have pushed the boundaries of what's possible in neuroscience and stem cell research. This research was made possible by support from the Tri-Institutional Stem Cell Initiative, the National Institute on Aging, New York State Stem Cell Science, the European Molecular Biology Organization, and several other organizations. Funding for basic science is crucial for discoveries like this, which can lead to new treatments and therapies. The ability to speed up neuron maturation in the lab marks a major step forward for neuroscience. As researchers continue to refine these techniques, we can expect faster progress in understanding and treating neurological diseases. The work at MSK is a reminder of how basic science can lead to breakthroughs that change lives. Stay tuned as scientists push the boundaries of what's possible in brain research.